Another episode of Study with Ash. For those of you who are new to the channel, I am currently on my second pass of first aid, and I'm really looking forward to study with you guys and help make revision easier for you all. So today we're going to cover two important topics. That is, the first one is the nucleotide structure, and the second one is the pyrimidine and purine synthesis. So we will start with the nucleotide synthesis. So what is a nucleotide? So there are two types of bases that are present in nucleotides. What are bases? So bases are either purines or pyrimidines, and they may have a two double ring structure like this or a single ring structure like this. So we need to remember that the nucleotides are composed of nucleosides plus phosphate groups. So nucleosides are equal to base, which is basically either a purine or a pyrimidine, and it is combined with a sugar to make a nucleoside. So once a nucleoside is formed, a phosphate group is added to the nucleoside by a 3' to 5' phosphodiester bond. We do not need to remember, we do not need to go into detail of this 3' to 5' phosphodiester bond, but we can remember it by the thing that this is mainly the direction of synthesis of bond formation. And the significance of direction is basically that the 5' end bears the triphosphate. So basically it has the energy source for the bond, right? Then we need to remember, so to remember what purines are, they have given us a mnemonic that is pure as gold. So I used to think of it as that I had a friend named Aisha, and I used to think that she's very pure, so she must be a purine, and Aisha is great, so purine is A and G, which has two rings as two best friends. Then we have pyrimidines. Pyrimidines is basically CUT. So pyrimidines, since they have one ring, they are small and cute. So you remember CUT, cytosine, uracil, and thymine as pyrimidines. I know this may sound like basic information, but sometimes you're sitting in the exam room and you might think, oh, was this a pyrimidine or a purine? Oh, I remember. Pyrimids are cute. Pyrimidines have one ring, and thus they are CUT, cytosine, uracil, and thymine. So I remember the pyrimidines. So these are some demyelination reactions that you don't necessarily have to memorize right now, as we will be going over some of these xanthine and hypoxanthine reactions later. But we need to remember that when cytosine is replaced by uracil, it is usually a spontaneous reaction, and you need to remember that this is repaired by base excision repair. And why is this important? Because uracil is not present in DNA. Alright, so we also need to remember that between the adenine and thymine, as A is comes earlier as compared to C, between adenine and thymine we have two hydrogen bonds, whereas between the cytosine and guanine we have 3 hydrogen. So for purine synthesis, you need to remember, if they say, so you are inhibiting purine synthesis pathway, you are inhibiting purine pathway, what amino acids would be affected? So they would give you one of these options, that you would get glycine, aspartate, or glutamine. Alright, glutamine has a lot of importance. Why? Because when glutamine is raised in cases of hyperammonemia, you get the signs of hepatic encephalopathy, and when you get the signs of hepatic encephalopathy, you characteristically get a flapping tremor. And questions would ask you what this flapping tremor is due to, and the answer would be glutamine. Normally this glutamine is converted into glutamate, but if there is a lot of glutamine due to hyperammonemia, you need to remember that it is basically toxic. So that's it from this page. We will be moving on to the next page now. That we have to, when we come to this page, we start thinking, oh my god, I would have to memorize this page. But that's not required for step 1. You need to remember that step 1 requires an understanding of how you link this biochemistry to actual way, how you prescribe immunosuppressive drugs, how you prescribe anti-cancer drugs, how would you prescribe antibiotics, and how they interfere with the nucleotide synthesis. That's what you need to know. But now you would say that for that you would need to memorize the steps, right? No. So basically what you need to remember is the rate limiting steps in biochemistry. And for this particular de novo pyrimidine and purine synthesis, you need to remember the enzymes. The two main enzymes that you really need to remember is the carbamyl phosphate synthetase 2 and the PRPP synthetase. So let's start with the pyrimidine based production. So what you need to remember is basically that you need carbamyl phosphate synthetase 2 as the rate limiting enzyme. So if you're looking about a question, so you're looking at a question and that says so you're getting large levels of carbamyl phosphate, then you need to think that you got the person might be taking leflunomide. So leflunomide basically blocks the dihydroorotate dehydrogenase, which is not even written here. So sometimes they do not write what's even required. So dihydroorotate dehydrogenase is inhibited by leflunomide, and this way you do not have uric acid. So however, if uric acid is formed and this is further not converted into UMP, you need to remember that this is uric aciduria. So repeat with me. Okay, I started with pyrimidine synthesis. I do not need to remember the entire pathway. What I need to do is, that I need to remember that the rate limiting enzyme of the pathway was carbamyl phosphate synthase 2. Okay, I remember that now. The second thing I need to remember is that in case my patient is taking leflunomide, that inhibits dihydroorotate dehydrogenase. So after carbamyl phosphate synthetase, I formed uric acid, and that uric acid particularly that is not further converted into UMP. So this is why I'm getting uric aciduria. Right? This is what I need to remember. So if you get carbamyl phosphate in your serum and the levels are raised, then you have blocked via leflunomide. But if you get uric acid in the urine, it means you have particularly blocked this particular pathway, right? Then the next thing is that if you give 5-fluorouracil, this pathway is blocked. This is important. So what you need to remember is these particular drugs and the rate limiting enzymes. Alright? So you need to remember that you need CPS2, you need PRPP2 here. Okay, मैंने carbamyl phosphate बना लिया. Then if I give leflunomide, I would inhibit dihydroorotate dehydrogenase. I would have high levels of CPS in my blood. Then I would, if uric acid is formed, leflunomide, there is no leflunomide, then what happens is that I would get uric aciduria. If uric acid is fine, it is converted into UMP, it is converted into UDP, but it is not converted into dihydroxyuridine diphosphate, DUDP. This means there is no reductase reaction, and UDP is not converted into dihydroxy-UDP, and this is due to inhibition of ribonucleotide reductase, and this is due to hydroxyuria. This is what I need to remember. I do not need to remember that CPS, uric acid, UMP, UDP. I need to remember if UD is not converted to UDP, I do not have ribonucleotide reductase. What blocks ribonucleotide reductase? Hydroxyurea, right? This is what I need to remember. Then once I form DUDP, then I have to form the dump. If dump is formed and DTMP is not formed, you need to remember that thymidinate synthase is inactive. So if thymidinate synthase is not present, N5 and 10 methylene tetrahydrofolate cannot be converted into dihydrofolate, and dihydrofolate is not further converted into tetrahydrofolate. And if it is not converted into dihydrofolate, then it means this is something that you need to remember. So if you're giving a person methotrexate, that what does methotrexate do? It causes bone marrow suppression. Why does it cause bone marrow suppression? It causes an overall decrease in synthesis. So why does it happen? This is because dihydrofolate cannot be converted into tetrahydrofolate. It cannot provide methyl group. It cannot provide the thing to make DTMP, right? It cannot make DTMP. So the overall synthesis is decreased. So you need to remember in this particular step that methotrexate, the dihydrofolate reductase is inhibited in this. DTMP is decreased in where we give methotrexate in cancer. So we are destroying human cells in methotrexate, right? They would also give you pyrimethamine in the same stem. You would be like, oh my god, they have the same mechanism of action. So what am I going to do? You need to remember that pyrimethamine blocks protozoa. P for protozoa, P for pyrimethamine. What about the third medication? What if it's present in the same stem? You would see, oh, they've given me trimethoprim. That's for bacteria. That's something we give as an antibiotic. So this is something that you must remember. This human bacteria wali cheese, this they ask in both UWorld and NBME, I guess. Then we have covered leflunomide pretty well. Then we have 5-fluorouracil. 5-fluorouracil, we remember that 5-fluorouracil was used in basically thymidylate synthase. So thymidylate synthase is, we remember, it is used to make DTMP from dump, and if it is locked, we have decreased levels of DTMP. Then we have the purine synthesis. For purine synthesis, if we start over here, from ribose, the phosphorybyl pyrophosphate is formed, and then it is converted into IMP. So basically this also has a lot of steps, but we need to remember that this is what we need to know. This is all what we need to know. So that's important, what we need to know. So 6-mercaptopurine, methotrexate, and azathioprine, these are some things that you need to know. So this blocks the conversion of PRPP to IMP. So it has an immunosuppressive effect and it concurrent use of xanthine oxidase inhibitors. So here, if you're using xanthine oxidase inhibitors, what happens? For xanthine oxidase inhibitors, you need to remember that, okay, enough of my girly stuff. You need to remember that 6-mercaptopurine conversion increases. So the reason the use of xanthine oxidase inhibitors exactly increase is basically xanthine oxidase inhibitors, they also break down 6-mercaptopurine into active metabolites, and these are toxic. So this leads to severe immunosuppression, and you need to reduce the dose and monitor the CBC levels, because this can lead to bone marrow suppression, giving a picture of pancytopenia. So never give 6-mercaptopurine, azathioprine, Basically, the azathioprine is product of 6-mercaptopurine. So you do not give xanthine oxidase inhibitors with them. You need to remember this drug interaction. Then we have mycophenolate and ribavirin. So this converts the IMP to GMP. So IMP dehydrogenase is inhibited. So all in all, you need to remember, first of all, the rate limiting enzyme CPS2. So CPS2, you need to remember that it is present in the cytosol. It is involved in pyrimidine synthesis. Whereas CPS1, you would study in the urea cycle, and that is involved in mitochondria, urea cycle, and it is found in liver. Right? CPS2 is found in most cells. So protein synthesis can happen in any cell, whereas urea cycle particularly happens in the liver. So overall, just a recap, you need to remember that for pyrimidine synthesis, the rate limiting enzyme is CPS2, which is present in the cytosol. Then you need to remember that if you use leflunomide, that is going to increase levels of carboxyl phosphate in your blood, and it is not going to form uric acid. Then if uric acid is not being converted into UMP, you get uric aciduria. If UMP is converted into UDP and UDP is not converted into dihydroxy UDP, you need to remember that you've inhibited the ribonucleotide reductase enzyme, which by hydroxyurea. And you need to remember that if dump is not converted into DTMP, you have inhibited thymidylate synthase by 5-fluorouracil. I know these drugs are tricky to remember, but I need you to know that if you revise this page for one day, then you revise it on the second day, then 10 days later you revise it again, and you do it via spaced repetition, there is no way you would forget this pathway. But you need to have your main focus on the drugs. Otherwise you would just be distracted by all the names. So that's it for this video. I hope you like it. I'm a beginner in this, so I would really appreciate any feedback from your side. Thank you so much.